Thanks, Julian. Can you hear me all right? So yeah, I'm Maya, and I will be talking to you today about the work we've been doing so far on what can we predict from the virus in relation to the host that it might infect and the transmission routes. So Julian mentioned in his talk that the first 100 days post-emergence, or the first 100 days of a pandemic, are very important. And that's because what we do within those 100 days determine progression of a whole pandemic or an epidemic. So if we put measures to stop movement of animals or movement of humans, for instance. However, I would argue that the 100 days is already too late. Actually, by the time a virus emerges as a human, it's already too late. And we must definitely prepare before that. So the work we've been doing aims to gain as much insight as we can, as early as possible, about what viruses exist in the world, what might they do, and what will they do, using data and machine learning. So our end game is that immediately after a virus is identified, say immediately after we get a sequence of virus, we want to understand and predict everything it might do, where it might go, and where it might hide. So where are we now in relation to our end game? So we have been working with what, well, not me, but humanity have been sequencing viruses since the early 90s. And we have been publishing papers about viruses and other pathogens for at least a century. And in the University of Liverpool, we have a database, and it's one of many databases available now where we have been collating this knowledge. We call it the Enhanced Infectious Diseases Database, the ID2, and we have mined all publicly available sequences that tell us this is the sequence, virus, human, etc. What it's been sequenced, what pathogen, what host it might have been sequenced from. And we have mined information from over 8 million publications. So while there are other resources out there, EID2 is the world's largest of pathogens, their host, and where they are in the world. So because we have accumulated this data, what can we do with them now? So if you look at the picture here, that was the state of viruses pre-coronavirus. So the sharing of viruses between humans, as you can see, we know a lot about humans. We know a lot about animals we eat and plants we eat. We know some information about primates because we use them in research. But out of that, we know almost nothing. However, this data is now a critical mass that we can do something with. And one of the things that we can do with this critical mass of data is predicting hosts. So what are the wild animals that are susceptible to known viruses? And why that is important? So particularly outside specialists, we think of a virus that will come from the wild, oh, it will come from a bat, infect humans. Great. But reality is very, very, very different. Viruses get around many host species, or not necessarily host species, but they circulate a lot in the wild. Sometimes they spill to humans, sometimes they spill back from humans, sometimes they just spill between animals and never reach human. And that is kind of a reality. And what we did is that taking our critical mass of data, we looked at this problem as and found out that given all the known viruses and all their known hosts, we only know 0.2% of interactions that might appear between those animals and those viruses. So we took that and we called it a network and said, out of those 99.8% gaps, how many of those might exist actually right now? And we don't know because we haven't sequenced the virus in the animal, or this animal is rarely studied and we haven't looked for this virus in them. And we call it real reality. But our approach has always been different because in computer science, there's a lot of network completion problems. But also, if you are a bioinformatician, you will look at the virus genome and say, okay, from the genome, I can predict the host. If you're an ecologist, you will look at the host site and say, okay, from the host, I might be able to predict which other animals might be susceptible to this virus. So what we did is that we brought all of those three, we call them perspective, together. And what we did is that we trained loads of classifiers in each of those perspectives. So if you imagine the host, assume it's human, we looked at all the viruses, whether they infect or not infect humans, and then we predicted based on that whether a virus would or not infect humans. If it's the virus, then we say, at the time of this work, SARS-CoV-2 was not yet a thing. So say SARS-CoV-1, which animals have been found as host to that, and which animals are not, and so forth. And then we brought all those models together, and we generated some results. And the reason why we wanted to do this approach is twofold. One is to enhance predictive performance, and the other is to enable explanation of our predictions. So if you see here, on the left is the host, the mammalian host and their viruses, and on the right is the network after our prediction. So we found that at least 20,000 virus-host association could potentially exist in nature, but we haven't found them yet because we didn't look for them. And we also showed that there is at least 4.3-fold underestimation of wild host of viruses. And if you can see here, because it works two ways, we can look at what characteristics or traits of the animal predicts its association with a certain virus, and we can also do it from the virus side. We also saw that there is 3.2-fold underestimation of the average mammalian host range. Great, so that's one about where viruses might hide. Where could they hide? So we can also look at geographically, where might they hide? And this is a work we've done on monkeypox, mpox virus, when the current epidemic first started. So the previous approach or the previous work looked at all mammalian viruses. This zoomed on pox viruses in both mammals and birds. And we showed that there is a potential, should the virus make it to European wild rodents, that it might establish itself in Western Europe. And you can see here how it also translates to all mammalian species. So what can we do with the critical mass of data we have? We can look at where might novel viruses come from. So we looked at predicting animal species in which coronaviruses might recombine. And we used, again, the same approach that we split the perspective, and then we looked at it and this is, for instance, where SARS-CoV-2 might recombine with other coronaviruses to create yet more SARS-CoV-3. And we found that there is underestimation in the host range of coronaviruses, including a 40-fold increase in coronavirus recombination hosts, so hosts that might be susceptible to 4 or more subgenres of coronaviruses. And again, you can see here how the nature might, well, if our predictions are correct, how reality might be very different to our observed state. So that is some of the stuff that we can do about, or we have done about the host range of viruses. Another thing we've been looking at recently is, can we develop machine learning pipelines that can predict the transmission route of a virus? So how the virus might be transmitted within a species or between species. So why is that important? Why is it important we know a transmission route of a virus? Because obviously that will tell us a lot of things that the virus can do. Particularly, like if you remember, at the very early days of the pandemic, there was a lot of debate of whether people should leave their groceries for 2 days before they touch it. And I know people who would just get it out, put it in the sun, leave it for 2 days, 3 days, then touch it again. And same thing applied to masks. So there was a lot of debate about, should we mask, should we not mask? So if we know these things early on, if we know how virus can be transmitted, we can then prepare to how we can mitigate against the spread. And another thing is uncovering the drivers of that transmission. So which parts of the genome might correlate with the virus being vertically transmitted, or fomite-borne, or vector-borne? And finally, also because we want to fill the gap in our knowledge. So a lot of the time, we know, for instance, if it's arthropod-borne, we'll stop. Okay, it's mosquito-borne. We don't look for any more roots. But as we've seen with Zika, it was mosquito-borne. Then we found it's vertically transmitted in humans. Then we found it was also sexually transmitted in humans. And the same applies to some of the mosquitoes. So some of the mosquitoes can sexually and vertically transmit Zika within their populations. So with that in mind, when I first started actually working on this, one of the things that popped up was, what is a transmission route? So I don't know if any of you work with plants, but don't try to put plant work and animal work in the same pipeline, because they don't talk to each other. So when I first started doing this, I spent time just reading papers from so many different places to just get my head around the terminology that is used in every field. And what we did is then identified 59 transmission routes and vectoring mechanism. And we captured those information about those for over 25,000 virus host association. I'll get to that in a bit. But what we've then done is we built this hierarchy, this big tree over there that shows how these routes interlink with each other. So if we look here, the size of the node tells you how many virus host association are associated with that route. So the vast majority, for instance, are horizontally transmitted, and very little is vertically transmitted. And then you can see how, for instance, for vertical transmission, the vast majority of it is what we call true, which is, for instance, if it's in plants, it will be via seed and pollen. If it's in animals, it will be within within mammals, versus semi-vertical, for instance, if it's outside on the shell of the egg. And that took a while, because obviously to capture this information, we have to mine lots of papers, read them again, go through textbooks to get the data. And then obviously we needed to map that to the host virus association, because I mean, there is no point predicting if a virus is the transmission route of a virus without its host, because a virus can have many, many transmission routes and deploy them differently to different hosts. So then we did that, and then we're like, what are we going to do with this data? And again, we used our multi-perspective framework because kind of like it, and it works. So from the virus side, we took the sequences, we took the full sequences, and we engineered lots of features from them, looking at things like biases in the codons, biases in the amino acids, but we also looked at some morphology as well, morphology information, and some basic genome stuff like GC bias and genome length, etc. So the vast majority of our predictors came from the virus side, but we needed the host, and that's where the problem happened. So when we looked at mammals, because they are well studied, we have a lot of information about them. We have lots of traits data, geographical data, life history, diet data, etc. But you go to birds, okay, there is some data there. You cross to other vertebrates, the data become fewer and fewer. You go to plants, good luck getting any data. So we did a proxy, and we just looked at divergent time between species. So that gave us a way to basically filter, because a virus transmission route is very dependent on the type of species it might infect. So a plant virus is going to deploy totally different routes to an animal virus. Similarly, a virus that infects mammals will deploy somewhat different routes from a virus that will infect birds. And then we also used our networks again. Don't worry too much. It looks like a matrix. It is not a matrix, but it enabled us to look at, so instead of just look at phylogenetic neighborhood between viruses, we looked at it with a network perspective and adding some similarity between the hosts. So we then put all this together in a predictive framework. Again, don't worry too much about the details, but if you have any questions, you can ask me at any time. So we put everything together we did some prediction, and the first thing we wanted to know is, can we tell a respiratory from a saliva transmission, for instance? What are the predictors of each transmission route? So if you look here at the green matrixy thing over there, what we found is that the features that we constructed from the network and the host side were very predictive of a transmission route for that association. However, the other features we engineered from the genome sequence enabled us to zoom in and generate more finer grained predictions. And I will get to this in a bit, but different viral features were also important for different routes and transmission modes. So for instance, if we look here, these are the predictors of direct transmission. And all you need to know is that each little dot here is a virus-host association, and the color is its route. So whether it's skin to eye contact, respiratory transmission, biting, or oral contact. And you can see, for instance, that the virus being enveloped is very important to predict if it will be transmitted through oral to skin or bloodstream contact. But the length of the virus is more predictive of it being respiratory, and also if it gets transmitted through skin and skin to eye contact. So as we can see, the virus sequence or the predictive features that we engineered from that sequence is telling us a different thing for every route and every transmission mode. Same here, if you look at arthropod-borne routes, you can see that certain biases in amino acids, for instance, hydroxyl, is very important for it being strep-borne or not, but it wouldn't affect it being midge-borne or not. So that, now we start to form a picture about what we can tell from the virus itself about its transmission route. And then what we did with that after is we had lots of virus-host association that we didn't have any information about their transmission. So there are viruses linked to humans, animals, plants, etc., that they haven't been studied yet for transmission route. So we put them through our pipeline and we started looking at what we see here. And if obviously we predicted more horizontal than vertical transmission, but we also start predicting more of vector-borne transmission within plants and more direct transmission, for instance, in animals, etc. So this is one application of this work, is that we can just now, we get the virus from the host, put it through the pipeline, it tells us how it might be transmitted to that host. And we also looked at, if you remember, we wanted to look at gaps in our knowledge. So we looked at our error or prediction within sample, and we looked, are they meaningful? Are those errors, or are they missing information? And if you can look here as well, we can see that there are some gaps of knowledge here that might, like we don't know all arthropod-borne transmission for all the viruses that we have some knowledge about its transmission route. And the final part about this work was we wanted to see if we can tell apart two transmission routes that are very similar to each other. So are we predicting, every time we predict a mosquito, are we also predicting it's midge-borne? Every time we predict it's airborne, are we also predicting it's fomite-borne? So we looked at it statistically and we found that that is not the case. All you need to know is that blue is good, red is bad, and we don't have much red. And that's good, because that's mean our predictions are not dependent on each other. So I gone very quickly through the work that we can do with the data we have right now. So given the amount of data that we have access to, this is very much what we can do. Obviously, there are other methods and other groups are working on similar stuff, but this is more or less the limits to what we can do with the data we have. But coming next, Marcus will also tell you about what we can do, how can we move beyond the limits of our knowledge so far. And with that, I'd like to thank you thank Marcus as well, because we work closely together, and collaborators and funders who sometimes refuse to fund this work. Thank you so much.
Something that is skin, for example.
Yeah.
So if I gave you the sequence of a papillomavirus,
Yeah.
would that then directly classify it as skin transmission?
It depends on the papilloma virus, and if it's very similar to ones that are skin-borne versus because obviously if it's different or not. And we'll tell you also the probability of it being skin-borne.
But have you run those through? Have you run like, so what is it? You have your training data and your test data. So have you run things through your test data to inform?
Yeah, and I mean, I think the skin-to-skin one was really highly predictive.
Right.
So the performance metrics were out, I didn't put that slide, but our performance metric varied a little bit between very understudied transmission routes, like, I don't know, weird ones in plants mainly, and much better for skin-to-skin respiratory arthropod-borne.
So before I hand to Simon and Miles, I was particularly taken with your idea of being able to predict coronaviruses that infect the same species, potentially the same cell, and potential for a combination, right? Can you then look at those coronavirus sequences and say, actually, if infectious bronchitis, a bird one, let's say SARS-2 can infect birds, in actual fact, they'd never get together because their sequences are so different.
Yeah.
Whereas if you put SARS-2 with a bat sarbecovirus, of course, they would be more likely. Can you bring that type of sequence information into it as well to rank them?
Yeah, we did not think about that. That's a good idea.
That's because you get rankings.
We should do that.
Yeah. Right. Thank you, Simon.
Thanks. Yeah, thank you for those beautiful graphical representations of data. I really appreciated that. Given that you've got such a large database already, is it too early for you to give us any red flags of concern or predictions on what might possibly be the next pandemic or cause for serious concern?
Well, yes and no. So Marcus will talk more about this, but given how biased the data we work with is, it becomes harder and harder to predict things coming to humans because of the gap. Like, we would know more after virus emerged in human. So for instance, we would not have imagined SARS-CoV-2 to happen at the time. But now the whole effort of humanity is focusing on coronaviruses to the extent that the next virus might come from a totally different family that we haven't yet even considered to study. And the same thing with when they linked it initially to pangolin. So one example I like to give, before the pandemic happened, we knew nothing about the virome of the pangolins. We had one virus. Within the first year, we got to know 6 viruses because people had pangolin samples in their fridges, and now suddenly they're publishable. Same with a lot of bad viruses. So now the bad data is becoming like very good because of the effort. But what happens if the virus comes from an animal we haven't even considered studying? And the cool thing about this is Marcus will talk a lot, right? About how we might be able to figure this. So if
and if you don't, you tell them.
If we put the two together, then yes, we will be able to predict the next pandemic virus. But I believe without putting the two together, we are still one step behind. So our work looked, sorry, just one more thing. Our work looked more about completing the picture in the wild side from what we know so far.
So really nice work, and I'm going to be a bit obnoxious now. So I sit on the WHO committee for predicting high threat emerging viruses, and we meet yearly in Geneva. The last time we met was 2018, and Christian Drosner we all had our predictions from all the epi data that's out there, and we came up with our top 8 pathogens. Obviously, pathogen X is always there. And Christian Josman said, well, considering the past and looking forward, would we not all agree that the next epidemic is going to be caused by a highly pathogenic human respiratory transmitted coronavirus? So, that's just logic. There's a lot of logic there. And so you fill in some gaps, which are great, but predicting what will come next, I mean, Christian was right on likelihood, and obviously flu is always on there. But it's being, for a virus, it's about being in the right place at the right time, and the international community not taking action. You have to look at that as well, to allow that initial transmission to expand to such an extent it's impossible to stop. So there are various non-computational elements to it as well. And tomorrow I'll talk a little bit about trying to predict finding out what is out there, and then tying it back into data sets like yours to give us sort of a risk profile. But it's when someone says, what's going to be the next outbreak pathogen, it's near on impossible to say.
Try to bring more than one perspective into the problem. So I think prior to pandemic, maybe not many people were looking at the full picture. Now there are more groups looking at it. But it's very important to look at, as you say, not just the virus, but the host and also the location.
Exactly.
Which is also one thing we're working on.
Thank you.